Assalamu alaikum students. Hope you all are fine and in good health. This is your online lecture of biology FSC part 1 from chapter number 13 immunity, and in this section we will talk about third line of defense. So students, here we will just summarize the three lines of defense, the way in which our body it responds to different pathogens or different microorganisms. So immune response, it is of three different types or of three different categories. First line of defense, second line of defense, and third line of defense. So we have studied previously about the first line of defense. That first line of defense, it is external. So when any pathogen it tries to enter our body, we are having some external barriers that will limit the pathogens out of the body, or that will not allow the pathogens to enter the body. Those external barriers, they are involved in case of first line of defense. So first line of defense, it is external and it is non-specific. And here we have studied about the role of the skin as an impenetrable barrier and mucous lining of the respiratory and digestive tracts. Then if first line of defense it fails to encounter the pathogens, or in whatever manner the pathogens they have tried to enter the first line of defense, now the second line of defense it is activated. Second line of defense, it is internal as it's inside the body, but it is also non-specific. So second line of defense is non-specific and it is internal, whereas first line of defense it is non-specific and it is external. So in second line of defense, we have studied about different killing cells of the blood, which may include the macrophages, neutrophils, natural killer cells, different types of the proteins, which include the complements proteins of the complement system and interferons. Then we have also discussed about the fever and inflammation. Now moving towards the third line of defense. Now if the pathogens they are not encountered, or the second line of defense it fails to encounter the pathogens, then third line of defense is activated. Third line of defense, it is internal and it is very specific. So students, we have to remember that third line of defense is internal and it is a specific response of our body immune system to a certain specific pathogen or microorganism. So this third line of defense, it is of two types: cell mediated immunity and humoral immunity, which we will discuss here in this section. Now here you can see the flow sheet diagram of these three lines of defense. Here you can see the first line of defense. These are the external barriers, which include skin and mucous membranes of respiratory and digestive tract. If barriers are penetrated, the body responds with the second line of defense, which is non-specific and internal. Second line includes phagocytosis. Phago means solid eating process of the cell. So phago means solid, and cytosis means with the engulfment of the cell membrane. So phagocytosis is solid eating process of the cell. Natural killer cells, they're also included in second line of defense. Inflammation occurs and fever or pyrexia occurs during second line of defense. Now, if second line of defense it also fails to stop the pathogens or to limit their growth, then the body responds with third line of defense, which is very specific immune response, and it includes cell mediated immunity and humoral immunity. So students, the third line of defense, it is specific resistance to any specific kind of antigen. So specific resistance is a third line of defense. Forms the immune response and targets specific pathogens. So the third line of defense, it is internal and it is very specific. It encounters the pathogens, specific pathogens within their own specific style or own specific manner. Specialized cells of the immune system, which are called lymphocytes, they are involved. So here the lymphocytes are involved, which are the types of white blood cells. These lymphocytes may include B cells or T cells, also termed as B lymphocytes and T lymphocytes. B cells, they produce specific proteins called antibodies, which are produced against specific antigens. So students, whenever any pathogen or any foreign particle it enters our body, that pathogen is having a certain specific protein on its surface. That specific protein is referred as an antigen. So in response to that antigen, our body it produces antibodies. So these antibodies, they will try to kill that pathogen. Then T cells are also there. T cells, they target pathogens directly. So T lymphocytes or the T cells, they will target the pathogens or the microbes directly. Now here in this diagram, you can see the third line of defense. We're having the role of lymphocytes, which may be B lymphocytes or T lymphocytes. B lymphocytes, they will help in the production of antibody or they will produce the antibodies, and T lymphocytes, they help in cell mediated immunity. Third line of defense consists of a specific immune response and is the body's main immune response. So students, we have to remember that the third line of defense, it is internal, specific, and it is the main immune response of our body to different types of the pathogens or to the specific pathogens. And this third line of defense, it is the most powerful means of resisting infections. Therefore, it is also called specific defense. It helps the body to build up a resistance and fight long-term infections. Third line of defense comprises of antibodies, T cells and B cells. So students, do remember that the third line of defense, it comprises of antibodies, which are produced in response to the antigens, T cells and B cells. Antibodies are proteins produced in the lymph that bind to specific antigens and destroy them. So as discussed earlier that the pathogens or the foreign substances, they're having a protein on their surface termed as antigens. So in response to the antigens, antibodies are produced within our body, and those antibodies, they are certain proteins that will bind to the antigen and destroy them. T cells and B cells are lymphocytes that are activated by the presence of antigens. So T cells and B cells, they are lymphocytes, or these are the WBCs which are present in our lymphatic system, and they are activated by the presence of different antigens. Third line of defense, it is the specific defense mechanism of the body. If a pathogen is able to get past the body's non-specific defense, the third line of defense interferes with a series of defense responses, often called immune responses, that attack the particular pathogens having specific antigens. So if whatever the source may be or whatever the manner may be, the pathogen it has finally escaped or it has finally crossed the first and second line of defense, then third line of defense is activated and it interferes with a series of responses, and those immune responses are to some specific type of the antigen. These antigens serve as the stimulus to produce an immune response. So the antigens which are present on the surface of the pathogens, they will serve as a stimulus or a change to produce a specific immune response. The term antigen, it comes from antibody generating. So antigen, this term comes from antibody generating substances. So these substances, in response to them, antibodies, they are produced within our body. Viruses, bacteria, and other pathogens have specific antigens on their surface. So different these pathogens, maybe viruses, bacteria, they are having certain proteins on their surfaces termed as antigens. Since the third line of defense respond against particular infections and acts as the most powerful means of resisting infections, therefore it is also called specific defense. Its response can be of two types: humoral immune response and cell mediated immune response. So third line of defense, it is very particular, it's very much specific, and it is the most powerful or most strengthening resisting infections, means of resisting the different types of infections. And there are two types of the third line of defense, which includes humoral immune response and cell mediated immune response. Humoral immune response, it is so named because the substances which are involved, they are found in the humor or body fluids. So in case of humoral immune response, the substances which are involved in them, they are found in humor or the body fluids. Cell mediated immune response, it can be defined as the activation of the T cells by a specific antigen. So in response to the humoral immune response or humoral immune response, it is carried out by the B lymphocytes, and cell mediated immune response, it is carried out by the T lymphocytes. So if we talk about the humoral immune response, it's has been given this name because the substances which are involved, they are found in the humor or body fluids. And cell mediated immune response, it can be defined as activation of T cells by a specific antigen. These immune responses are particularly carried out by two components: B lymphocytes or B cells and T lymphocytes or the T cells. The third line of defense, it also involves role of monocytes as well, that participate in activation of these lymphocytes. So as we have discussed earlier that monocytes, they are having role in second line of defense. The monocytes or the macrophages, they are also having a role in third line of defense. So monocytes, they are the WBCs, and when monocytes, they leave the blood stream or they leave the blood, then they are referred as macrophages. Macro means large, phage means eater. So macrophages, they are large eaters which will carry out the process of phagocytosis. Next we are having, so we will just start with the role of monocytes in third line of defense. As we have discussed in the previous part, that in second line of defense, monocytes are kind of white blood cells or WBCs which are produced by the lymphoid tissues. So our lymphoid tissues, which may include spleen, maybe the thymus gland, they produce certain white blood cells which are referred as monocytes. These monocytes, they circulate in blood for 10 to 20 hours, and ultimately they leave the blood and come into the intercellular space of tissues. So monocytes, they will continuously circulate, or they will be circulating within the blood round about for 10 to 20 hours, and afterwards they will leave the blood and they will come in the intercellular spaces of the tissues which are termed as interstitium. In the tissues, they will swell and attain a larger size to become tissue macrophages. So when the monocytes, they will leave the blood, they will attain more size or they will increase in size, and finally they will turn into macrophages. So monocytes, When they leave the blood, they are termed as macrophages. When macrophages perform phagocytosis of invaded microorganisms after digesting them, they not only display microbial antigens on their surface, but also begin to secrete about 100 different compounds, including various enzymes, interferons, and a protein called interleukin 1. So students, we have to remember the various functions of the macrophages. That macrophages, they will carry out or they will perform phagocytosis. Phagocytosis means the solid eating process of the pathogen. So the macrophages, they will just encounter the pathogens and they will engulf them, digest them. Secondly, not only they digest those pathogens, but they also display some of their parts on their surfaces. So they will display microbial antigens on their surface so that the other cells can also recognize that the antigens are present in that area. So they will display microbial antigens on their surface as well. Thirdly, they also secrete different types of the compounds, which may include enzymes, interferons, or different proteins. And a very specific or important protein which they produce is interleukin 1. So students, do keep in mind that macrophages, they will perform phagocytosis, they will engulf the pathogen, digest it. Afterwards, it will also display the parts of the antigen on its surface. And thirdly, they also produce different important compounds, which may include enzymes, interferons, and proteins. And one of the most important protein is interleukin 1. The interleukin 1 secreted by macrophages activates the T cells that in turn begin to secrete interleukin 2, which then activates the B cells. So students, do remember this flow sheet, that the interleukin 1, which is produced by the macrophages, or we can say that macrophages, they will secrete a protein termed as interleukin 1. That interleukin 1 protein, which is secreted by macrophages, they will stimulate or they will activate the T cells. Then in turn, the T cells, they will produce interleukin 2, and that interleukin 2, it will activate the B cells. So macrophages will produce interleukin 1, and from those interleukin 1, the T cells, they are activated, and from the activated T cells, they will in turn produce interleukin 2, and from this interleukin 2, the B cells, they are activated. Interleukin 1 also promotes a general response to injury, causing fever and activating other mechanisms that defend the body against different pathogens, or that defends body against invasion. So interleukin 1, it also promotes injury. It also promotes response to the injury, which may include fever and inflammation. Next, we are having the role of T cells in third line of defense, that is cell mediated immune response. So students, do remember that the role of T cells, it will carry out or it will bring about cell mediated immune response, whereas the B cells, they will bring about humoral immune response. So first we are having the role of T cells in third line of defense or cell mediated immune response. T cells, they originate from stem cells in the bone marrow. So T lymphocytes or the T cells, they are originated from the stem cells in the bone marrow. Stem cells, what are the stem cells? They are the basic cells that can become any type of cell in the body. I'm repeating, stem cells, they are the basic cells and can become any type of cell in the body. They basically come during the embryonic conditions, or maybe during the adult conditions as well. So stem cells, they have got the potential to become any type of cell in the body. So T cells, they are originated from stem cells which are present in the bone marrow. After early embryonic development, or during the embryonic stages, the newly forming T cells, they migrate to the thymus gland for processing. So the T cells, they are formed within the bone marrow, and from the bone marrow they will migrate to the thymus gland for processing. Thymus gland, it is a lymphoid organ. It is located behind the sternum and lungs, and it has role in lymphatic system. So T cells, they are originated in stem cells in the bone marrow. From the bone marrow, after early developmental conditions or early embryonic conditions, they will move towards the thymus gland, and in thymus gland their processing will be carried out. The thymus makes T cells immunocompetent, that is capable of immunological response. So until and unless the T cells, they migrate to the thymus, they are not able to uh, give any immunological response. So when they move towards the thymus gland, the thymus, it makes the T cells immunocompetent, means that they are competent of producing certain immune response. The immune response provided by T cells is called cell mediated immune response. So students, do remember that immune response which is provided by the T cells is cell mediated immune response, and immune response which is provided by the B cells is humoral immune response. Now, how the activation of T cells is carried out. In case of infection, macrophages perform phagocytosis of invaded microorganism and detect particular antigens, non-self molecule of the organism. So first of all, what will happen if any pathogen, it just enters the third line of defense, then macrophages, they will perform phagocytosis, or they will engulf those pathogens, they will digest them, and this all occurs because they detect particular antigens or particular proteins which are present on the surface of those pathogens. Macrophages isolate and display these antigens on their surface with the help of their own proteins or the self proteins called major histocompatibility complex. So when the pathogens, they just reach the macrophages, or macrophages, when they detect any pathogen, they will recognize those pathogens, or they will detect particular antigens which are present on the surface of those pathogens. Then they will ingest the pathogens, they will phagocytose them, and they will digest them. Finally, they will isolate some of the parts of antigens and display them on their own surface. That display, it is carried out by their own proteins, and those proteins, they are referred as major histocompatibility complex. So with the help of MHC, or major histocompatibility complex, the macrophages, they will display some parts of antigens on their surface. In this way, macrophages become antigen presenting cells. So now the macrophages, they are termed as antigen presenting cells because they are presenting some parts of the antigens on their surface. Macrophages also secrete interleukin 1 that stimulates and attracts helper T cells towards the displayed antigen. So macrophages, as we have discussed, that macrophages, they will also produce a protein termed as interleukin 1. This interleukin 1, it will stimulate or activate the helper T cells or the T cells towards the displayed antigen. So now the helper T cells, they have specific receptors on their cells termed as T cell receptors. So because of those T cell receptors, they will bind to the macrophages, or they will bind to the antigens. Macrophages, they also secrete interleukin 1 that stimulates and attracts helper T cells towards the displayed antigen. So now here you can see the activation of cell mediated immune response. First, here we are having a macrophage. This macrophage, it will detect the antigens which are present on the surface of microbe. Finally, it will engulf that microbe. It will digest it or phagocytose it, and then it will display some parts of these microbes or some parts of these antigens on its surface. As here you can see this triangular structure, this is displayed on the surface of this macrophage, and this displaying is carried out by own proteins of the macrophages, which are termed as self proteins. So these self proteins of the macrophages, they will help to display parts of antigens on its surface. Now, the third role of the macrophage is that it will produce a protein termed as interleukin 1. So firstly, do remember macrophages, they will carry out phagocytosis of microbe. Then secondly, it will display some parts of the antigens on its surface by the help of its own proteins termed as self proteins or major histocompatibility complex. Thirdly, they also produce a protein or they secrete interleukin 1. This interleukin 1, it will stimulate or it will activate helper T cells. Now helper T cells, they recognize the antigen because they are having some specific receptors. Here with this blue color, you can see these specific receptors. These specific receptors are termed as T cell receptors. So because of the T cell receptors, helper T cells, they will bind to the displayed antigen. Helper T cells have specific receptor on their surface called T cell receptor, by which they bind with a particular antigen displayed on antigen presenting cell. As we have discussed that when the macrophage, it will present some parts of the antigens on its surface, now it is referred as the antigen presenting cell. So finally, the helper T cells, they will bind to the antigen which is displayed on APC or antigen presenting cell. Interleukin 1 also stimulates the helper T cells to secrete another protein, the interleukin 2, that not only compel the helper T cells to divide but also causes the proliferation of certain cytotoxic T cells and B cells. So, the helper T cells, this interleukin 1, it will help helper T cells to bind the antigen, and it will also activate the helper T cells. Now these helper T cells, they will produce another protein which is termed as interleukin 2. Now this interleukin 2, it will stimulate certain cytotoxic cells and B cells. The activation of T cells by a specific antigen is called cell-mediated immunity. The body contains millions of different T cells, each able to respond to one specific antigen. So students, we have to remember that the activation of T cells by a specific or by a particular antigen is termed as cell-mediated immunity. And our body, it has got millions of different T cells which respond differently to specific antigens. So we will just memorize this diagram again, that activation of cell-mediated immune response, how it occurs. Macrophages, they will recognize the antigens on the surface of the microbe, then they will engulf them, digest them, and they will display some parts of these antigens on its surface with the help of their own proteins. These you can see over here with green color, and these proteins are referred as self protein or major histocompatibility complex. Now when the macrophage, it will display these proteins on these antigens on its surface, now they are referred as antigen presenting cell. Now macrophages, they also produce a protein termed as interleukin 1. Now due to this interleukin 1, which is secreted by macrophages, helper T cells, they are activated, And helper T cells, they will bind to the antigen because the helper T cells, they are having a receptor on its surface termed as T cell receptors. Now here you can see that helper T cell, it is having the T cell receptor, and this, by the help of this T cell receptor, helper T cell, it will bind to the antigen. Now helper T cell, it will in turn produce interleukin 2, and due to the interleukin 2, B cells and certain cytotoxic T cells, they are activated. Now here you can see the mode of action of cytotoxic T cells. Cytotoxic T cell binds to infected cell. Here you can see that this is a cytotoxic T cell. It will bind to the infected cell, because the infected cell, it is showing the parts of the antigen on its surface with the help of its own protein called non-self complex self proteins, and this complex is now referred as self non-self complex. Now cytotoxic T cells, they will produce certain cytotoxins or certain perforin molecules which will cause some pores in the membrane of this infected cell. Now here you can see that perforin makes holes in infected cell membranes and enzyme enters. So cytotoxic T cells, they will produce perforin molecule, and these perforin, as the name indicate, they will form certain holes or pores in the membrane of this infected cell, and as a result, the enzymes, they can enter the cell. Finally, the enzymes, sugars, or other substances, they enter the cell. As a result, the size of the cell, it increases, and finally the cell ruptures, or it bursts. So now moving towards certain types of the T cells, there are two main categories of the T cells, which include CD8 cells and CD4 cells. So the first group is known as CD8 cells because they have surface marker designated CD8. So CD8 cells, they are having certain tags on their surface or certain surface markers on their surface which are referred as CD8. So that's why, because the CD8, they are having surface markers called CD8 on its surface, that's why these are referred as CD8 cells. CD8 cells, they include cytotoxic T cells and suppressor T cells. Second group is of the helper T cells is also known as CD4 cells because they are having the surface markers termed as or designated as CD4. So CD8 cells are having surface markers called CD8, and CD4 cells, they are having certain tags or surface markers on their surface called CD4. When T cells are activated, they divide and produce four types of cells which have specific role in cell-mediated immune response. So T cells, when it divide, it produces four different types of cells. First, we are having cytotoxic T cells. As the name indicates, cytotoxic T cells, they produce a toxin which is referred as cytotoxin. This cytotoxin, it triggers or it speeds up destruction of pathogens DNA. So cytotoxic T cells, they will produce cytotoxin, and cytotoxin, it will cause destruction of the pathogens genetic material or destruction of the pathogens DNA. Then cytotoxic T cells, they also produce perforin molecules. This perforin, it is a protein that will create holes in the pathogen's plasma membrane, and as a result, that pathogen, it will burst or it will lyse. Next we are having, so do remember students that cytotoxic T cells, they produce cytotoxin and perforins. Next we are having helper T cells. Helper T cells, they will secrete interleukin 2, which stimulates cell division of T cells and B cells. So as discussed earlier, that in turn the helper T cells, they will secrete a protein referred as interleukin 2, and interleukin 2, it will stimulate T and B cells. In other words, we can say that these cells recruit more army, or these cells, they recruit more cells to fight the pathogen. Next, we're having the suppressor T cells. As the name indicates, Suppressor means to suppress. So when an infection is successfully removed, the suppressor T cells, they will suppress the immune response. So these cells begin to secrete certain proteins that inhibit or that stop further proliferation of T cells. So suppressor T cells, they will produce some proteins which will stop further multiplication or further proliferation of T cells. Therefore, suppressor T cells, they will suppress or they will shut down or stop the immune response. The fourth one is memory T cells. Now, as the name indicates, the memory T cells, they will keep a memory with them that this type of antigen, it has entered the body previously. So these cells remain dormant, means that these cells, they remain inactive after the initial exposure to an antigen. So after the initial exposure or first time exposure to any antigen, they will remain dormant or inactive. If the same antigen presents itself again, even if it is years later, the memory cells are stimulated to convert themselves into helper T cells and fight that pathogen. So when that antigen it is again given or it again enters the body, the memory T cells, they're having the memory and they will stimulate themselves to become the helper T cells and they will fight the microbe or that pathogen. So students, today we have talked about the third line of defense. The third line of defense, it is internal and it is very specific and it is the most powerful response of the body. Then third line of defense, it is of two types: humoral immune response, which is due to B cells, and cell mediated immune response, which is due to the T cells. Then we have also talked about the role of the monocytes in third line of defense and role of T cells in third line of defense. So good luck students for memorizing the topic. This was all for now. Till then, a very happy learning. Thank you and Allah Hafiz.